Welcome to Blue Oak Nutraceuticals, science-based, heart-led. Today, we're in conversation with Dr. Sundeep Dugar, a drug discovery and development expert and co-founder of Blue Oak Nutraceuticals. Sundeep will talk about the causes and consequences of mitochondrial dysfunction and how exercise can have a positive effect. The following slides reference clinical studies involving the mitochondrial supplement MitoCatalyst. These studies were conducted on specific individuals in a limited population setting for limited periods of time. Any references to these clinical studies is not intended to be representative of results that should be expected from the use of MitoCatalyst for anyone in the general population. These statements have not been evaluated by the Food and Drug Administration. These products are not intended to diagnose, treat, cure, or prevent any disease, and children under 18 and individuals with a known medical condition should not take this product without first consulting a physician.
Good morning, everyone. First, let me apologize for not being there in person. Unfortunately, my travels preclude that possibility. Today, in the next 10 minutes, I'll try and touch about the relevance of mitochondria in mtARS. And unfortunately, we won't have time for questions and answers. But if there are any ensuing questions, I'll be happy to connect again and discuss those with you. So just a brief primer on mitochondria. Next slide. So mitochondria, as we all know, are the essential components of our cells. They are the only organelle in your cell that can make more of itself when needed, fix itself when broken, and get rid of itself when not needed. And that's an aspect you should keep in mind about its ability to do these things as we go further into our discussion today. Concept of biogenesis is how the mitochondria create more mitochondria. They have a half-life of about 28 days, so your body naturally makes more mitochondria to replenish the ones that are eliminated from the cells post their half-life. Mitochondria are also can be destroyed, or they face destruction through the formation of permeability transition pores, which are caused by exogenous insults or high cellular calcium levels, glucose, ischemia, etc. Now, the main purpose of mitochondria and fundamental to life and health is they take in nutrients and turn it into energy. We call it ATP. ATP is now the storage form of this energy, and they fuel all cellular activities. Just some tidbit about that. You have about 100,000 trillion mitochondria. About 25% of your cellular weight is mitochondria, which means that each cell has between 1,000 to 2,500. And some cells in certain organs, high energy demand organs, have even more mitochondria per cell. And at every function and every activity of a cell of the body, the ATP is associated with it, and the mitochondria hence has an influence on it. Now, a healthy person at rest produces their body weight in ATP every day. And at maximal exercise, this can increase to 500 to 1,000 gram or 1 kilo per minute. That tells you the rapidity at which mitochondria function to supply bioenergetics to a cell. And the total amount of ATP needed is 10 to the power of 25, which you can just imagine is a huge amount that's needed every day. What this implies is that the role of the mitochondria is critical, quintessential in life and health. Next slide. Just how did the mitochondria come about? And it is an interesting idea that the concept of the mitochondria, or the process in which cells make energy, is ubiquitous and universal to every living form on planet Earth. Every animal species, plants, and aquatic animals, they all use the concept and engendered in the mitochondria to produce this energy. And scientists have hypothesized that somewhere about 1 to 2 billion years ago, there was an endosymbiotic event that led to the amalgamation of a aerobic bacterium or a cyanobacterium into an ancestral prokaryote cell. And eventually, these ancestral eukaryote cells split up to provide the cyanobacterium, or the green part, became part of the plant kingdom, and the aerobic bacterium became part of the animal kingdom. So, in essence, mitochondria are a bacteria. They have their own DNA, and they reside outside the nucleus, the nuclear genome. Next slide. So, in essence, mitochondria are essential to all biological functions, as I've said to you before. I list here just brain, muscle, liver, and health as an example, but I could easily have stated the whole human body, and that applies to your skin, your hair, your eyes, everything require a mitochondrial function to be optimally functioning. Next slide. Mitochondrial DNA is inherited only from the mother. So, as I mentioned to you, it has its own genome. It is a bacterial genome. It's a circular genome, as shown on the right side of the slide, and it only comes from the egg. In the time of conception, the mitochondria in the sperm is destroyed, and the surviving mitochondria is only from the mother. So, while the nuclear DNA has generational changes, the mitochondrial DNA does not. It comes directly from the mother's lineage to her mother, to her grandmother, and so on. Next slide. So, having two genomes in your cell, the nuclear DNA and the mitochondrial DNA, you can imagine there are two mitochondrial pathologies. One is associated with primary mitochondrial diseases, where the mitochondrial DNA has a mutation in it, causing dysfunction in the mitochondrial processes. And the second is a secondary mitochondrial disease, where the impact, or the exogenous or the endogenous insult, is outside the mitochondria, but the mitochondria feel the stress, the cellular stress associated with this, that leads to secondary mitochondrial dysfunction and depletion. So one is the causality on the left, and one is the consequence of something else on the right. mTARS resides on the right. It is a secondary mitochondrial disease and not a primary mitochondrial disease, and due to the associated mutation in the synthetase. Next slide. So then what happens? You have a consequences of mitochondrial depletion dysfunction, and that spans the entire physiology of your body and can manifest itself through these multiple phenotypes, whether it's metabolic syndrome, such as diabetes secondary to prescription drugs, sarcopenia, associated with also loss of muscle strength, muscle atrophy or disuse atrophy, heart failure, neurodegeneration, chronic kidney disease, and dot, dot, dot. So it goes on. And that presentation of the pathophysiology is often related to the genetic profile of the patient. Let's talk about a little about mitochondrial heteroplasmy. So, in a situation of a secondary mitochondrial pathology or secondary mitochondrial disease, cells contain both healthy and unhealthy mitochondria. And this is termed mitochondrial heteroplasmy. So that the cells have normal working mitochondria, at the same time they have a dysfunctional mitochondria, which implies that the cell already have a bioenergetic deficit that they are working with. And that leads to the mitochondrial dysfunction and depletion. Cells cannot be completely mitochondrial null because they would not be alive. So for the cells to be alive, there has to be sufficient mitochondrial bioenergetics in the system. But it's the deficit that leads to this, or the chronic insult leads to this deficit that leads to the physiological presentation. And this is something I borrowed from the mtARS website. And you can see the effects of that is related to developmental delays, ataxia, hypotonia, so on and so forth. And as I mentioned before, a lot of these are also presented in other diseases that also have secondary mitochondrial myopathy. So what this is suggesting is that a mitochondrial deficit or dysfunction as a secondary pathophysiology, even though the primary insult is different, the eventual pathophysiology starts to be the same because they all go through the mitochondria of the cell. So the primary insult may be different, but the follow-on secondary pathophysiology is the same because it comes to mitochondrial depletion and dysfunction. So this presents a reasonable association of an approach that addresses mitochondrial depletion and dysfunction to have a beneficial outcome, at least to the phenotype or the clinical presentation of the disease in patients. And this is something we have seen before in other studies, but we think that this particular association has a significant value. Next slide. So here we have, let's deeper into mitochondrial heteroplasmy. So if you look at the left, in patients that either have secondary mitochondrial myopathy or disease or primary, you have both functioning mitochondria in open circles and dysfunctioning mitochondria or mutated mitochondria in the red. And as these cells divide, the cells divide disproportionately to generate a healthier daughter cell, which means that there is a less healthy daughter cell. So you can see that in one daughter cell, you have 20% of the mitochondria that are dysfunctional, but on the other, they become 60%. As this divides again, you can get to 40% and 80% proportional separation. And as the number of dysfunctional mitochondria increase, that leads to the clinical manifestation. So now the cells on the top right have 80% dysfunctional mitochondria, and they present the clinical phenotype that people see. And so, and what is that? It leads to accumulation of depolarized and ROS generating mitochondria, attenuated ATP production, improper cellular metabolism, and signaling pathways. And as these things happen, the consequences and the damage to the mitochondria continue, and the attenuated ATP production leads to all other secondary clinical physiology onto the as presented by the patient. So, next slide. One thing we have known is that exercise improves mitochondrial function, and it can have a positive impact on managing symptoms of mTARS, and it has been shown, such as it improves muscle function, strength and endurance, it reduces fatigue, improves aerobic capacity, reduces pain and stiffness. And the associated rationale for that is, and it has been shown that increased energy demands during all exercise stresses mitochondria and induces pathways that activate mitophagy, primarily through the pink one, the parking pathway. And why is that relevant? Because mitophagy is a process that cleans up old or dysfunctional mitochondria, and then that can lead to improved mitochondrial quality and function, better ATP production, etc. What that means, as I've shown you previously, that when you have a high percentage of dysfunctional mitochondria per cell, and you have an exercise-induced uh, impact on these cells have the ability to induce mitophagy to remove those damaged and dysfunctional mitochondria, because it's also an inducement related to available bioenergetics. As the cells are depleted in bioenergetics, instead of inducing mitophagy, thus allowing these dysfunctional mitochondria to accumulate, the cells are prioritizing the use of that energy for critical body functions, such as organ functions of the heart, the brain, metabolism, and any other residual critical function. So the body and the cells are making a choice in the absence of reduced bioenergetics to forego mitophagy. And it has been shown that with exercise, these pathophysiologies or these effects of the mtARS are improved, or the consequences are reversed. And the argument has been that it is due to improved mitophagy, improved cellular bioenergetic function. So I hope my message has was clear. Unfortunately, when I'm not in person, it's not the same. But hopefully, at another time, we will have an opportunity to discuss this further. Thank you for your time, and look forward to talking about this more.
Thank you for joining us today. Learn more about Blue Oak Nutraceuticals and MitoCatalyst at www.blueoaknx.com.